Hello everyone, my name is Zebang Chen. Today I would like to introduce a paper published on ACS catalysis. The name is Reversing the Entantial Selectivity of Enzymatic Dynamic Kinetic Asymmetric Transformation in the Synthesis of Fused Lactones. This research group is in the Sichuan University. In this chapter, our topic is about protein engineering. So the protein engineering generally refers to the modification of amino acid sequence of a protein to optimize its stability, activity, specificity, solubility, or other functions through the regional design or directed evolution. So in this paper, the following two methods of the protein engineering are mainly used. The first is semi-rational design, the SRD. This is not a random mutation, but targeted mutations of two key sites according to the enzyme structure and substrate binding mode. These two amino acids are located in the active pocket of the enzymes and are closely related to the substrate binding conformation. The second is a function of the target modification. So the enantiomer selectivity is regulated by the alternating the substrate binding conformation through the Bug polarity or pi-to-pi interaction of amino acid residues. The same citrate can be selectivity synthesis with two enantiomer selectivity products by two different mutants to flip the selectivity, and the shift from the kinetic spinating, the Kr, to the dyKT, the dynamic kinetic asymmetric transformation, can be realized on some substrate to turn the reaction mechanisms. And I would like to introduce a background. So the importance of the fused lactones. The fused lactones are a class of important structural units widely found in nature products, drug molecules, and flower molecules, especially in the field of chemical biology and fine chemicals. Among the eta-lactones are widely used as nature flavors in food, flavor, and tobacco industries, and the sterol structures of these compounds play a decisive role in their functional performance, especially the conformation of multiple consecutive chiral centra directly affects their odor recognitions and biological activities. The significant sensory difference between their enantiomers suggests that the higher selective synthetic methods are important for precisely modulating the properties of fused lactones and the challenges of existing synthesis method. So common strategies for the synthesis of chiral fused lactones including the asymmetric reduction of the substrate to obtain a chiral hydroxy ester or acid intermediate followed by lactonization to form the target products. These strategies theoretically has the advantages of a high atom economy and clear pathways. However, for bulky substrate with a dense chiral centers such as tricycle and these strategies still face many challenges. So the first is the complex design and synthesis of the chiral ligands, and the high dependence on substrate structure and poor applicability, and they often require the harsh reaction conditions such as high temperature, strong acid environment, and it is difficult to flexibly regulate the enantiomer selectivity of the products, especially difficult to realize the reversible enantiomer switching. Therefore, there is an urgent need to develop a green synthesis method with high selectivity, mild conditions, and enantiomer modulations in order to solve the sterol selectivity problem in the construction of fusion lactones. So in this research, their objects consist of four parts. The first is about the construction of the keto reductase mutants with inertial selectivity complementary. And second is about development of high selective enzyme catalytic KRAR and DYKT strategies. And third is about expanding the range of the catalytic substrate for efficient synthesis of multiple fusion lectons. And last one is analyzing the enzyme substrate recognitions and selectivity mechanism by molecular simulation. So firstly, the researchers comparatively use comparative strategies for fusion lecton synthesis and regional enzyme designs. So this part on the FIGO-A shows some previous existing methods, such as lactonization with a Cooper catalyst, which can produce the lectons. But the reaction temperature is high and the conditions are harsh, or dynamic kinetic splitting and hydrogenations with the residue catalysis, which can avoid the yield lectons. And although the catalyst cost is high and the stereoselectivity is very dependent on the catalysis structure, there is also a class of the carbon injection reaction involving the P450 enzymes, which is a distinctive approach but very restrictive in terms of substrate type and experimental conditions. And we can look at the FIGO-B. It is an innovation of this article. So the structure of the BCHE8220 keto reductase was designed by protein engineering, and two complementary mutants, the M3C3 and M4A1, were created by changing only two sites, the H145 and Y188, which can control the enantial selectivity of the alpha keto ether substrate with great precision, and one of them has a preference for the ARBS products and one for the ARBS products. And the BS products are one of preferred to generate the ASBR products. What's more, these two enzymes can not only do kinetic splitting, but also realize a dynamic kinetic asymmetric transformation on specific substrate, which means that no matter whether you give it a R or S body, it can turn to a substrate into the chiral products you want. You can also see through the structural simulation that the two mutants have different active pocket configurations leading to different substrate binding, so which can determine the chiral configuration of the products. The next part is about determining the enantial selectivity and activity via the targeted mutations in BCHE8220. So this figure shows how the researchers systematically screen the mutants by the structural modification of the BCHE8220 to achieve the precise regulation of the yield and the enantial selectivity of the fusion lactone products. So let's start with the first two graphs on the left. So in the graph A, the researchers use the M2 mutant as a starting point for the saturation mutagenesis at the key site Y188. So it can be seen that most of the mutants are weak in the kinetic ability with a yield below 10%. But there is a totally 4 specific mutants show the clear advantages, such as Y188C, Y188G, Y188N, and Y188A. So they can achieve the yield up to 49%, and it shows the importance of the YDA locus in the regulation of the activity. And in the FIGO-B, so it shows the effect of different mutation combinations on the enantial selectivity of the products. It can be clearly seen that the single mutants such as M3C prefer to generate the 3AR9BS configurations, while when the YDA mutant was combined with H145 mutation, such as M4A1. So ethane energy of the products was successfully flipped to the 3AS9BR. So we can realize that the ethane enantiomer selectivity controls. And finally, in the left table, in the right table, the author listed the reaction results of all mutants, including the yield, the enantiomeric ratio, and the diastereomeric ratio. And several of the mutants, such as M3C4, M333, M4A1, shows outstanding performance, not only the high yield, but also with completely opposite enantiomer selectivities. And the DR was also greater than the 99 to 1, showing the very excellent performance and the modulation ability. So next, they discuss about the broad substrate scope of the enantiomer complementary K-red mutants. So this figure shows how well the two complementary mutants constructed by the author and react on different substrates. So all these substrates are structurally different. The Y ketoester, and the goal is to achieve the reduction and cyclization to tricycle fused lactones via the K-red catalysis. So each cell in the figure represents a substrate and gives the yields and enantiomeric ratio and the diastereomeric ratio of two enzymes on this substrate, respectively. So we can see the two remarkable conclusions. So first, in terms of the yield, both mutants have the good efficiency for most substrates, and especially on some substrated the aromatic rings and the heterocyclic ring and halogen modified substrate. But both these two mutants will show the yield around 50% to 80%, showing a strong substrate adaptability. And second, their enantiomer selectivity are clear complementary, with the M3C4 performing in the ARBS configuration, M4A1 flavoring, so preferring the ASBR. For example, on substrate 16E, M3C4 gives a enantiomeric ratio of the 91 to 1, whereas the M4A1 is exact opposite, the 1 to 99. So author calls this enantiomer selectivity reversal, which is very real and very useful. And next, they do the molecular docking simulation, reveal active site interactions, governing enantiomer selectivities. So this figure illustrates how the researchers explain the difference in the molecular mechanism of enantiomer selectivity between the M3C3 and M4A1 mutants by molecular simulation analysis. So in the figure A, so it shows several possibilities substrate binding conformation proposed by the researchers. So in 1 and 2, so we can find that R2A and S2A bind to enzyme activity pocket in different mutants in a favorable conformations, and it can accept the hydrogen activity ions provided by NADH, as well the protons provide the TYROH and resulting in the conformation of the stable cis products such as 1AS2R or 3R or 2R or something stuff. And in the contrast, the conformation in figures 3 and 4, there are two special hidden label as sterically hydrant for the substrate to be correctly localized in the pocket. It results in inefficient reactions. This is also explained why the certain transconformation products were not detected, as these conformations are unstable or cannot form in the enzymes. And you can find in the figure B, So it shows the results of author molecular dynamic simulation or the structure of the four complex. So in the BA, it's about the M333 bind to the R2A with the substrate in the proper conformation. So this with the distance between NADH and the substrate only the 3.4 and 4.0. So it's facilitating the hydrogen transfer. And in the FIGO 2B, B2, when M333 binds to the S2A, the substrate is 4.8 away from the hydrogen donors, so which is not favorable for reduction. And in the FIGO B3, the M4A1 pairs with the S2A at a very short distance. It's only 1.9 and showing an optimal catalytic conformation. But when it binds to the R2A and distance of the 4.5, it will due to the spatial crowding and can result in the decrease in the catalytic efficiency, which is shown in the FIGO 4B. So this simulation clearly explains why the M333 prefers R2A and M4A1 prefers S2A, which is a molecular basis for the innate selectivity of two enzyme mutants. And this is the last part is about the kinetic profiles distinguished reaction pathway in enzymatic lactone synthesis. So this graph shows researchers' time-dependent monitoring of substrate consumption and products generation during the reaction of two different substrates, used to determine whether the enzyme catalytic mechanism is KRAR or DYKT. So the figure on the left shows the concentration changes curve when the substrate 2A is involved in the reaction. So in this system, the R2A is rapidly consumed, while the concentration of S2A remains almost unchanged. While the products 3AR and 9BS 2E accumulates, so this trend suggests that the enzyme is only active only one of the enantiomers, and that the S body is largely unresponsive. So the reaction will follow the classical KRAR mechanisms of the kinetic splitting and asymmetric reduction. And the figure on the right, it shows another substrate, the 18A. So in this system, both enantiomers R18 and S18 are consumed simultaneously, and corresponding products will continue to rise. So this suggests that both are efficiently converted during the reaction and consistent with the substrate undergoing a rapid keto process, and thus the reaction is follow the DYKT mechanisms. Well, follow these mechanisms. Furthermore, they do the mechanistically elucidation of the enzymatic stereoselectivity in fused lactone synthesis. So, in this part, it shows that the researchers' speculations and validation of the mechanisms of catalytic reaction of these classes of enzymes, including the electrons transfer process, the mechanisms of the intercalations, and the confirmation of the pathways by deuterium labeling experiments. So in FIGO A, it shows the reaction mechanisms model proposed by the authors. So the RACA is a substrate which first undergoes ino-keto interconventions in the catalytic pocket of the enzymes to form the intermediate state A, which is supplied with protons by TYR156 and hydrogen negative anions by NADH to complete the asymmetric reduction to generate the trivial intermediate CISF, which is ultimately catalyzed into the products CISE in methanol. And the FIGO B, it shows that the deuterium labeling experiment performed by the author using the deuterated isopropanol with the substrate RAC88, which is after 40 hours of reaction and catalytic by the M333 mutants, produced the products 18E9BD with deuterium labeling of up to 94%, while the maintaining more than 99% enantiomer selectivities. In verifying the hydrogens or the deuterium is derived from NADPH, and that the reduction reaction pathway is valid. So in the last figure, so in figure C, it further compares the difference between the KIR and the DYKT mechanisms. So first is about the DYKTA process. So where it is able to interact rapidly with the RACA, so both enantiomers are converted to the same products. And the other one is KRAR. So when the interconversion is very slow, so that only one enantiomer participates efficiently in the reaction, while the other remains. And last one, I'd like to talk about the conclusion and the novelty of this research paper. So in this paper, they have developed an efficient and high selective biocatalytic method for synthesis of the tricycle fused cis-lactones using a modified BCHE8220 enzyme, which can be realized by either DYKT and KRAR reaction pathways. A pair of the sterol complementary mutant was obtained by the target mutants of only two key sites, the 188 and 145, allowing the precise control of the stereo selectivity of the lactones produced in the reduction reaction. And in the KIR reactions, not only the high enantiopure lactose products, but also the unreacted enantiometric ketones ester substrate was obtained simultaneously and shows a good kinetic splitting ability. And both the computational simulation and experimental studies show the source of the stereo selectivity is closely related to the spatial conformation of the residues and site of the 188 and 145 in the active pocket of the enzyme, and that the regional design of this region can be effectively regulate the binding poses and the conversion pathway of the substrate in the reduction process. And the difference between the mechanisms of these two will mainly depend on whether the substrate can form stable enantiomers under the enzyme catalysis. When the enol formation is easy, the reaction favors the DYKT process.